Welcome to Learn with Sam. Today we are going to deal with the topic Mendel's law of inheritance as well as the difference between monohybrid and dihybrid cross. So without any further delay, let's get into the topic. The first law is law of dominance. When two homozygous individuals with one or more sets of contrasting characters are crossed, the allele characters that appear in F1 are dominant and those which do not appear in F1 are recessive. The meaning for this law is when two homozygous parents are crossed, the character that appears in the F1 generation is considered to be dominant and the character that doesn't appear in the F1 generation is considered to be recessive. Consequently, the gene that codes for the character being expressed is considered to be dominant, and the gene or allele that codes for character which is not being expressed is considered to be recessive. Let's understand this law with the help of an example. So, in this example, we have taken two parents, a green pod and a yellow pod. Green pod has a genome, that is genotype capital G, and yellow pod has a genotype small g. Capital G codes for green color of the pea pod, whereas small g codes for yellow color of pea pod. So, we have two homozygous parents, capital G with genotype capital G and genotype small g. So, when we cross both the parents, we will get the following offsprings in F1 generation with the following genotype: capital G, small g, and small g. So capital G both are having dominant characters, so the color of the pea pod will be green. In heterozygous condition, that is the genotype capital G and small g, capital G is considered to be dominant and therefore green color is being expressed by the F1 generation. In the genotype small g, there is no dominant character. The both are recessive character or recessive trait because the genotype itself is recessive. Therefore, the color of the pod will be yellow. The second law is law of segregation, also called as law of purity of gametes. The statement is when hybrid F1 generation forms gametes, the allele segregates from each other and enter in different gametes. The gamete formed are pure in that they carry only one allele each, either dominant allele or recessive allele. Hence, this law is also described as law of purity of gametes. They never blend or mix with each other. They always segregate during gamete formation and enter into individual or different gametes. The third law is law of independent assortment. The statement is when hybrid possessing two or more pairs of contrasting factors alleles forms gametes, the factors in each pair segregate independently of the other pair. This law is based on dihybrid cross. It describes how different gametes or alleles present on separate chromosomes independently separate from each other during formation of gametes. These alleles are randomly united in fertilization. Now let's take a look at this with the help of an example. So for example, we have taken a dihybrid cross. So the parents are pea plant, one with capital R, capital Y, that is round and yellow seed, and small r, small y, that is wrinkled and green seed. So we have taken two sets of characters, two different characters. One is the color and second is the shape. So in color there are two, yellow and green, and in uh, shape we have round and wrinkle seed. So when we cross them, the alleles, the genes separate from each other and they randomly unite during gamete formation and fertilization respectively. The alleles segregate from each other randomly during gamete formation and unite during fertilization randomly. There is no hard and fast rule that capital R should unite with small y or capital Y should unite with small r. They can unite with any other allele present in the chromosome. Now let's see the difference between monohybrid and dihybrid cross. So the first point of difference is it is a cross between two individuals taking a single contrasting trait at a time. whereas dihybrid cross is a cross between two individuals taking two contrasting traits at a time. So the basic difference between monohybrid and dihybrid crosses, in monohybrid cross we will take one character into consideration, whereas in dihybrid cross we will take two characteristics into consideration. For example, in monohybrid cross we will take the character as height of the stem. So it can be tall or dwarf. So height of the stem is a single character we are taking into consideration. In dihybrid cross we have to take two characters. First is shape and second is color. Just an example, color of the pod and shape of the pod. So in color it can be yellow or green, and shape it can be round or wrinkled. So the next point is, it helps to in monohybrid cross it helps to study the inheritance of a pair of allele. In dihybrid cross it helps to study the inheritance of two pairs of allele. So monohybrid cross is helpful to study inheritance of one pair of allele. Dihybrid cross is helpful to study inheritance of two pairs of allele. The phenotypic ratio in F2 generation in monohybrid cross is 3:1. The phenotypic ratio in F2 generation in dihybrid cross is 9:3:3:1. The genotypic ratio in F2 generation is 1:2:1. The genotypic ratio in F2 generation in dihybrid cross is 1:2:2:2:4:1:2:1:2:1.